Question 5, page 38. And this is the blood typing question. You're going to see lots of questions like this on your midterms, or sorry, not your midterm, but your final, and of course on your unit exam as well. So this is kind of a go-to question. They really like this type of stuff here. So let's go through the question here. It says in human blood types, or for human blood types, the alleles for types A and B are co-dominant, but both are dominant to type O alleles. So if you remember in the student note packages, we said this is some of the nomenclature that you do have to memorize. A lot of times you're making up your own, you're giving nomenclature, arbitrarily giving them their own letters and stuff like that. But this one you want to memorize. So what that means is if I have a capital A and a capital B together as a genotype, they are going to be codominant, and I'm going to have AB blood type. I'm going to have those on my red blood cells. Those. So if we take a look at IA, doesn't matter what this other allele. Now it can't be a B, but that could be an O allele or another A. It could be homo A or hetero A. That's going to be A blood because A completely dominates over the O allele. Okay, and B, same thing for that. If I have an IB, doesn't matter what the other allele is. Now it can't be an A, otherwise that would be AB blood because those are codominant. But if it was hetero, so a little I, that's still going to be B blood because again, the B allele is dominant over the O allele. And the only way that I can have O blood is two little i's. And for whatever reason, that's the way that they show the nomenclature. That's the way you have to memorize it. So two little i's, because they're recessive to both A and B, I need both those presents before I'll have O blood. Now this is still a dihybrid cross because now they're asking about another trait altogether. So we don't just have ABO blood typing, which is one trait, but we also have something that's called the RH factor. Okay, and that's completely separate from the AB blood type. That's what it says in the question there, located on a separate chromosome. And it's saying that RH positive allele is dominant to RH negative allele. So if I have RH positive blood, I don't care what this other allele is, that's going to be positive blood. So you can have either A positive, B positive, or O positive. If I have two recessive alleles, Rh negative, Rh negative, that's going to be negative blood. And again, you can be AB negative, A negative, B negative, O negative, or whatever. Okay? But those are completely separate traits. So two traits, dihybrid. So here's a question, a little bit tricky, but we're going to show you best strategy, best practices. That's our legend there. It says indicate the possible phenotypes of mating a woman with type O blood who's Rh negative. Didn't mean to cross her out like that. With a man that is type A Rh positive. And you're going to see why it's just possible. So underline the parents. Let's talk about the genotype. So the woman, we'll just put the woman down here. If she's O blood, we can see by our legend she must have two recessive O alleles. And if she's Rh negative, same thing for that. Rh negative, Rh negative. Both are alleles. So we can actually determine her full genotype based on the fact that she's recessive for both those traits. But the male, so she's crossing with a male who is A blood. So if he is type A, look at our legend, he must have a capital I, capital A, but we don't know his other genotype or his other allele, sorry. We don't know if it's homo or if it's hetero. So what we're going to do with this type of question, because they're not showing us any babies here, so we can't go look at offspring and fill in this blank. So what they're looking for are the possible phenotypes. So what we're going to do here is we're going to make this line a little bit bigger, and we're going to put possibly what that other allele can be for him still to be A blood. So he could be homo A, meaning this other allele could be a IA, or this is how we're going to do it. Again, best practices, he could be hetero for A with a little I. And any of those examples are possibilities. He is still going to have A blood. He is either going to be homo A or hetero A. Now, he's also this for RH. So he's Rh positive. So we know if he's Rh positive, he must have at least this. But his other allele, extend the line a little bit, we know he could be homo positive or he could be heteropositive. with a negative. Okay, so now that we determine that, again, you can't go look at offspring because there are no offspring here. What they're asking for are the possible phenotypes of these, of their babies, right? Of mating these two parents. So this is how you kind of do this question. So bit tricky here, but often when I'm face to face with guys, I'll get them to use their fingers. So I'm going to show them that if I brought, now all this female can contribute is a little I. But this male, if he contributes the A allele, we know that one of the possible phenotypes of this offspring can be A blood, right? And if we take this negative, that's all she can contribute, and we take his positive, we know this baby could be positive blood. So that is one of the options. A positive is one of the possible phenotypes of the offspring with these two parents. Now, what I'm going to try to do here is, and I'm not great with technology, but I'm going to try to change my ink color here now. Let's make it green because we're going to show some other possibilities here now. And normally I get students to do this with their fingers. So all she can contribute is her little I, right? In her egg, that's all she can contribute. If he contributes, let's say, and it doesn't matter which one, let's put this IA. We know that baby's going to be A blood again, but take a look at this. This baby, if she contributes a RH negative, which that's all she can, and we know that one of his possibilities for his RH factor is RH negative. Well, if he did have that and he donated that in his sperm, we know that baby could be A negative. So that is another possibility. Now this gets pretty confusing when you're actually drawing out the lines, but normally we'd be using our fingers and it makes it much easier to figure out. So let's see if we can change our color again, our pen color again, and make this blue now. Now, again, all she can contribute is her O allele. I was hoping that color would be different. So I want the pen and I want the color now to be blue. So all she can contribute is that O allele. Now take a look at his possibilities here. We know he's A blood, but we said under the underline he could be homo or he could be hetero A. So if he is hetero A, and again we don't know, that's why it said what are the possible phenotypes, we're going to say what happens if he was hetero and he is carrying a recessive O allele. Well, then it's possible for him to have O blood. And that could be O positive, just like we determined already, or that could be the other option, O negative, right? So if this one contribute here, that's all she can contribute here, you could also have an O negative. Now, could they ever have an AB offspring? And of course, the answer would be no. For you to have an AB offspring, one parent must contribute the A, and one parent must contribute the B. But that's impossible with a female who's O blood. She can't do that. So you could never have an AB offspring. And these are the 4 possibilities just using our fingers. No, got a little dicey here with all these different lines. But if you just bring your fingers together, simulating fertilization, you can come up with this without any Punnett squares. Now, I have people that say, well, can I do a Punnett square? Sure, you could for this question, but look how many Punnett squares you'd actually have to end up doing. You'd be doing about 4 Punnett squares, take you forever. And once you do it with this way, just using your fingers and that, you can ascertain the answer much quicker. Probably if I wasn't talking, you could do this question in less than 5 minutes. Okay, a little tricky. Any questions? Give me an email and we can go over it. Thanks, guys.